Well, hi everybody. Hopefully you can hear me. Welcome to EnviSMART this month. My name's Brad Clark and I'm an academic from the University of Melbourne. At the start of the meeting, we'd like to acknowledge the traditional owners on the country that we're meeting and pay our respects to their ancestors, the elders past, present and emerging. With EnviSMART, we're able to keep this event open and free to anyone who'd like to attend each month, and we can do that thanks to the support of our supporters. That includes Agilent and Eurofins, and we've partnered with organisations, the Australian Contaminated Land Consultants Association, Water Research Australia and CTAC Australasia and Asia Pacific. And we also have partners with Trajan and PM Separations. So if you're not a member of some of those organisations, go and get your membership sorted out. They do good things. Each month we've got our program set for the rest of this year. We've got just continuing on with the great line of speakers we've had and we'll have today as well. So these are our upcoming talks. Next one we've got Michelle Hadlick from the USGS joining us, which will be fantastic. Jenny Storber talking about deep sea mine tailings, and then we have a bit of a plastics theme. We're going to have the amazing Jen Lavers from the University of Tasmania, and also Professor Kevin Thomas from the University of Queensland joining us. That says May, that should say September. So, but anyway, I'll update that while in the meantime. Okay, so without further ado, we'll get on to our presentation today. Caroline works at the Dutch National Institute for Public Health and the Environment as a Senior Policy Advisor on Pharmaceuticals and the Environment. As a European Registered Toxicologist and CTAC Certified Risk Assessor, she's worked on the environmental risk assessment of veterinary and human pharmaceuticals since 2006. And she's been involved in the development of a number of guidelines for this risk assessment. She's also a member of a number of expert groups on the European policy level and work package lead of the EU Horizon Premier project. Within RIVM, she also works or has worked on projects related to safety within the circular economy, drinking water and the Water Framework Directive to facilitate a more transparent and systematic evaluation of ecotoxicity data. She developed the CRED method. Well, without further ado, thank you, Caroline, and very much looking forward to hearing your talk today.
Okay, thank you, Brett, for the great introduction, and welcome all of you at my talk. For me, it's still quite early in the morning. For you, it's an afternoon. So I hope that after this, you've got some nice evening activities planned. Let me start sharing my screen. You should be able to see my presentation now. Is that correct?
Yeah, we can see and hear you very well. And thank you for joining us all the way from Europe the first thing in the morning as well.
I told my two sons who are sick at home at the moment that I was giving a presentation in Australia, and they were like, huh, what? Well, and that's of course because of the after COVID, we got all these nice systems and it's become so easy to do this. So thanks for inviting me, and I'd like to show you the stuff we did around the CRED method, where the main goal was to improve the quality of ecotoxicity studies and the use of ecotoxicity studies in regulatory frameworks. And that's what my presentation will be about. So first, what is the National Institute for Public Health and the Environment? That's a governmental body with the mission of a healthy population living in a healthy environment. We do research and advice for the government, and we are independent of the government as the policy makers, so we are policy advisors, and our independence is laid down by law. So if the policy makers ask us a question, they may not get the answer they like, because we base our answers on facts and on science, and they sometimes base their ideas, of course, on political agendas. So that's where there's a split. We're also not in the same town. There's a physical and physical and a non-physical distance between us and the policymakers. We are, however, owned by a ministry, and that's in this case the Ministry of Health, Welfare and Sport. We have 15 centers, 1,800 employees with 3 divisions. One is living in a safe, clean and healthy environment, and that's where I work. The second one is preventing and controlling infectious diseases, and that's division has been really busy around the COVID crisis. And the third division is good healthcare and a healthy lifestyle. Who am I? I think I can skip this one quite quickly because Brad has already told you. So then going to the contents of this presentation, as you're probably all aware, is that there's thousands and thousands of chemicals on the market, and a lot of them are not tested. Some of them are in the process of being tested, and a couple are actually approved by regulators based on usually the information by industry. But it's quite a huge task to do this safety assessment of chemicals regarding human safety and environmental safety. I'm an environmental risk assessor, so what I'll be talking about today is mainly environmental risk assessment. And if you then zoom into environmental risk assessment, there's many different types of ecotoxicity studies that you could perform to assess safety of a certain chemical or product. And these ecotoxicity studies, they have differences in their study design, in their methodology, in their quality, how they're performed. If you look at research lab studies, their level of detail reported is much higher than what is average in scientific literature, for instance. So all these different types of ecotoxicity studies, they all have their own merits, but they also all have their own flaws. And the regulatory use of published data, of peer-reviewed data in scientific journals or other open data sources is sometimes limited. Sometimes that's because the guidelines specifically say you only need an algae, a daphnia, or a fish. But even if a guideline says you should use all information, usually not all information out there is actually used. Many companies that perform their assessments focus on GLP and OECD criteria. Also, many regulators, and especially the regulators that are not very knowledgeable. For instance, I come from the pharmaceutical framework, where there's only a couple of European member states that really have the knowledge to look at the articles in scientific literature. Many of the member states, it's a non-clinical trial expert who also does the environmental side on the side, and they don't really have the knowledge, so they just stick to what they know. And that means that they focus on these research lab studies, which would then exclude most data in the public domain. And I'm sure that you know that there's a lot of interesting studies out there. So what does that lead to, in my view, is that we need a systematic assessment of reliability to help people actually use all these studies in the public domain, and that each study also should be evaluated in the same way, in accordance with scientific principles. So it doesn't matter if it's a research lab study or a published study, the same rules should apply to both. And that means that when it's not a GLP study or not performed according to an OECD guideline or a US EPA guideline, it may still contain very valuable information, especially when we're talking about non-standard test species and substances that have specific mode of actions. And my own personal practice shows that even these contract lab study reports with their OECD study protocols and GLP stamps may not always be reliable. We have seen enough studies that have not been performed correctly or not been reported correctly, where the contract labs try to hide sometimes in their in their data that something went wrong and they didn't want to spend a lot of money on reperforming the study. Another issue that is really important is that currently there's many different substance frameworks and many different people evaluating within these frameworks. So we've got REACH for industrial chemicals, we've got the plant protection products, we've got the biocides, veterinary pharmaceuticals, human pharmaceuticals, cosmetics, and so on. And these all have their own frameworks, their own guidelines, their own rules. Within Europe, 20 plus member states evaluating studies that leads to very different conclusions sometimes. And when we're moving towards a one substance, one assessments protocol, of course, that's not what you would like. So this is very important within the EU chemical strategy that we're trying to harmonize these evaluations. And of course, the impact of decisions may be large. If a substance, for instance, would be seen as a risk within the veterinary pharmaceutical framework, it could be refused marketing authorization. But if then a similar study or the same study for the same active substance, I mean, there's active substances that are a veterinary pharmaceutical as well as a human pharmaceutical, as well as a plant protection product, as well as a biocide. For instance, think of permethrin. It could be a PBT substance in one framework and not in another framework, or a critical study may be seen as reliable in one framework and not in another framework, which will influence the decisions made within those frameworks, and in the end influence what will happen with one of these products. And again, this harmonization to me is very important, and that has actually also been the onset of thinking about the CRED system, that we were talking about a specific product that was authorized in a number of frameworks could have been a PBT product, and then one framework wanted to take it up and the other framework said, well, for us it's not PBT because we look at it differently. And I'm like, well, how can a substance be persistent bioaccumulative toxic PBT with you guys with a total ban, and not with you guys and nothing happens? So this harmonization really is very important. And to start with harmonization, then, the basis of all environmental risk assessments is the data that you need and that you use. So what I would like to see is that all data out there is actually used, and that the data is evaluated in a consistent and transparent way, because if it's evaluated in a transparent way, it can also be used in other regulatory frameworks. And of course, you then need to put these evaluations also in a database. So if you have a database for industrial chemicals or a database for pharmaceutical products, then if a study has been evaluated, that should be clear, so someone else can just use those evaluation results and doesn't have to do it his or herself. And on the other hand, also, if someone doesn't agree with the evaluation results, that should also be clear, and then you could have a good focused discussion on what you don't agree about. So what's the general framework when you're assessing studies for ecotoxicity is that you start with a problem formulation. So what is my assessment? What am I going to do? What is then the evaluation method that fits with that assessment? Or is it a prescribed evaluation method in a guideline? What are the data available? Is this data generally usable? So when you do your Scopus search for all literature out there and you get 1,000 hits, of course you do a first inventory of all these hits where you delete all the studies that you can see already immediately that they don't fit what you'll be doing. And then you're going to assess reliability, and you're going to assess relevance of these individual endpoints in the study, followed by, in some frameworks, a weight of evidence analysis, and then your hazard or risk assessment. And today we're going to be focusing on the two aspects in these red ovals. So that's the reliability and the relevance of these studies. And what is then reliability? I'll be using that word a lot today. Reliability is the inherent quality of a result of an endpoint in a test report or publication. And I'm talking about that result or endpoint because in some studies you could have different sub-studies or different substances that are studied or different organisms that are studied, and sometimes the results are valid for part of what is reported but not for everything. So the test in itself is usually not the thing that you're putting your reliability assessment to. It's the results that are reported in that test. So the reliability is the inherent quality of that result that relates to a clearly described experimental design to allow for the study to be repeated independently. So it needs to be clear what someone has been doing. It also relates to the way the experimental procedures were performed. Did the study setup also meet the criteria for your compounds that you study? For instance, if you have a really volatile compound, did you use a system which is not open to air? Did you measure it enough? And then, of course, reporting of the results. So did you report your raw data? Did you show clear graphs? Are the graphs and the tables in accordance to each other? So then you can have some evidence, some proof, some statistics on is this correct, what has been done here? And the other part that has to be evaluated is the relevance. And the relevance is the extent to which data or tests are appropriate for a particular hazard identification or risk characterization. So the relevance depends on the purpose of your assessment. And that you can see in that figure to the right is that you have a lot of information, what you want to say, and there's the question that you have, and only part of your information is then relevant to the question. So here it's more graphical. You have your information, you have the purpose of the assessment, and only part of that information could be relevant. So where the reliability is an intrinsic quality of a study, and it doesn't matter what you use the study for, it will still be reliable. Whether you can actually use it depends then on the relevance to the purpose of your assessment. And that usually is based on the protection goal. And I'll give you a couple of examples. That also means that if you have a study which is really reliable and really relevant, for instance, for the derivation of a PNEC or a water quality standard, it may not be relevant for another goal or another assessment, for instance, when you want to develop soil criteria. So, for instance, here we've got a toxicity study with a terrestrial plant which is exposed to a herbicide in aquatic medium. That sometimes happens, that also terrestrial plants are tested in aquatic medium to see whether or not they grow. Is this then relevant for the derivation of soil quality standards? And the answer is usually no, because it's exposed via water and not seen as a relevant route. But then, is it relevant for the derivation of aquatic quality standards? And again, the answer is no, the species is not relevant for water. So the result of that toxicity test may be pretty reliable. The way it's been performed by using the wrong species in the wrong medium makes it not relevant for regulatory risk assessment. It may still, however, be a very reliable study and a scientifically very interesting study, but then we cannot use it for regulatory purposes. Another example, we've got relevant poster results with a study of an insecticide to an insect, to a midge, but it's a poster and we've got very few experimental details reported. Can this then be used for aquatic risk assessment? And the answer in this case is no, because it's a poster with almost no experimental details. So the reliability of this will be really low. It's, however, really relevant. So usually we're like, oh, we would like to use this so much. In some frameworks, if it's very relevant, they will allow it to use it as supporting evidence or in a weight of evidence approach. But of course, the first step in these cases is try to contact the author for more details in order to increase the reliability of that study. And usually when we're talking about posters, the authors will work on scientific articles, so you can ask the author to provide you the article as soon as it's available, for instance. When you want to evaluate reliability, and I'm fully aware that you cannot read this table, that's a table from the publication that is mentioned below. There's many methods that you can use to evaluate reliability of test endpoints or of studies. And these evaluation methods all have their own methodology, their own criteria, and their own way of how they are set up. So if you want to have an overview of all these methods, please look at that publication. Because when you have to choose a method to evaluate reliability, there's quite a number of points to consider. So for instance, are you going for categories, yes or no? Or do you want to give a numerical score from, for instance, from 1 to 100 or 1, 2, 3, 4? Do you want there to be criteria that are exclusion criteria? So like, if this is not met, that study cannot be used. Or critical criteria, this criterion should be met for this study to be used. Do you want the criteria to be weighed? Like this is a more important criteria than the other one? Or do you not like this weighing because you feel that every study could be different and thus these criteria, weighing them beforehand, might not be a good approach? Do you want the system to be tested on case studies? What it is applicable to? Is it only aquatic ecotox studies, or is it also broader ecotox studies or even human tox studies? Do you want it to have the standard guidelines incorporated in the methods? Do you want to have 15 criteria or 60? What kind of criteria do you feel are important for your framework? Do you need guidance, or can you do it without guidance? Do you need a web tool or an Excel tool, or do you have your own tool in your framework? So there's many points to take into account when finally choosing a method to evaluate reliability. And I think there's no right or wrong here, as long as you know what you're doing. Many people use the Klimisch method. The Klimisch method was published in 1997, so it's already been out there for 25 years. And Klimisch was the one that started with defining reliability categories in 4 different categories. So 1 is reliable without restrictions, 2 is reliable with restrictions, and I'll come back to what that means later. 3 is not reliable, and 4 is not assignable, so then you don't know. This study has no guidance, or very limited guidance, and it's often debated whether this could be biased towards GLP or OECD studies, as it has been published by someone from BASF, so it's been published by industry, and it leans pretty much on GLP OECD study protocols. These reliability categories, as promised, so there's reliability 1, reliable without restrictions, and that means that all critical reliability criteria for this study are fulfilled. Study is well designed, well performed, and does not contain any flaws that affect the reliability of the study. So this is just a study that you can use, that's well performed, that's good, put it in your database. Reliability category 2 is the category where the study is generally well designed and performed, but has some minor flaws in the documentation or setup. That doesn't mean it's not reliable, so it's still reliable, it can still be used, but it's not as perfect as that first category. In most regulatory frameworks, at least in Europe, both these reliability scores are used for risk assessment. And then there's reliability 3, not reliable. That means that not all critical reliability criteria for the study are fulfilled, and the study has clear flaws in study design or how it was performed. So this is a study where, for instance, there are issues with the solubility of your test compound, or with the controls, or with the statistics, or well, whatever could have happened to make a study not reliable. And then there's the study that is not assignable, is where you don't have the information to make an assessment. So for instance, this is a literature review where you just have the endpoint, or very limited information like that poster. So you don't know whether this study has been performed in a reliable way. And sometimes you can find out by looking, if it's a review, going towards the original source. Sometimes that source cannot be retrieved, or you have other difficulties there. And sometimes you can even see, when you go back to the original source, that there's been some recalculations done, and then you also don't know whether or not you can believe the figure that has been reported. So that not assignable is a very difficult category. Usually, if you get the information, then it could move up to reliability 1 or 2 even. So not assignable is definitely not the same as being not reliable. A not assignable study could become a reliable study if you would have that information. If you have enough information to see that it's not reliable, that you can already see the flaws in the setup, then it shouldn't be an RI4 study. It should already be assigned to be not reliable. I hope that's clear. So to sum up, we've got a couple of categories, and I'd like to make some more remarks about that, although I've already made some, so there's a bit of repetition here. It's important to note that reliable and reliable with restrictions studies are not only guideline and GLP studies. So peer-reviewed studies performed in laboratories could also be really reliable studies. If it's well prescribed, well performed, it may be a reliable without restrictions even. While on the other hand, even GLP labs sometimes report very poorly, sometimes do not look at their substances correctly, and then it's usually a matter of it being performed poorly, not exactly according to the guideline, although they make it look like it, because if they would perform it according to the guideline, they would not accept, for instance, issues with solubility, or it's not reported correctly. But we do see also GLP labs sometimes reporting not reliable studies. So be critical on that. That unassignable category is to be assigned only if a study lacks the details to evaluate reliability. Please contact the authors for more information. But if you are an author, this also shows that if you include more details on the study setup, and that you can now do that in the supporting information that everybody can then also find online, that will increase reliability and reproducibility. And in the end, of course, will also facilitate your review process. If the reviewers have a more clear view of what you've done and what the quality of your work is, then the chance of your paper being accepted increases. In practice, of course, I've been a bit negative about these contract laboratory studies, but in general, these studies are well reported, well performed, according to official guidelines. And of course, these types of studies are really easy to assess because normally all information is in there, and normally these studies are okay. So when you're an assessor, you usually like to assess these studies because it's all there. And in the peer-reviewed publications, you sometimes really have to search for the information to see whether or not it's reliable. I'm sorry, I've got a bit of a cold. So what's also important to remember is that a well-written study is not per se a reliable study. So you have to evaluate it based on the details provided. If it reads nice, it reads well, it's a good story, it may not, it really may not be reliable still. But of course, if you are a good writer and you have well-structured reporting, it does make things easier for the reader, and again also for the peer review process. So for you as students, please note that systematic reporting is also important. When we're performing reliability assessment within these regulatory frameworks, just the statement that the study is performed according to a certain guideline is not enough. As within these OECD protocols, there are still some degrees of freedom. So you want to see what has actually happened. So then even a study report that looks good from a scientific laboratory, accredited laboratory, but no details, just a statement, it has been performed according to this protocol, will still be then not assignable or R4. And again, what I said, if there are clear flaws in the study and more information will not change those flaws, you can assign reliability 3 to such a study, even if you don't have all information. But if you can already see, well, this and this went wrong, and whatever they've done with all these other stuff, it will still be a bad study, then more information won't change that. And these two, not reliable and not assignable, are often confused. So that's really important to keep an eye on that. So that is all more or less an introduction to the CRED project. So what did we do with the CRED project is we wanted to increase the consistency and transparency in the evaluation of relevance and reliability of aquatic ecotoxicity studies. As you've seen in my former slides, there's many things to take into account. You can mix up these reliability categories. So if we provide a systematic way of evaluation, the chance of these evaluations amongst member states or also between regulatory frameworks being harmonized is much higher. With this aim, we also aimed to improve the usability of peer-reviewed literature for substance evaluations, because we saw so much nice information out there that's not used because we cannot assign it a reliability score, because we miss information or because it has not been performed exactly, some details have not been performed correctly that could have been really easy to solve when setting up the study. And as I said, we also wanted to increase the exchange of assessment results between frameworks. So this is the paper as it was published in 2016. It's an open access paper, and I did this together with colleagues from Switzerland and Sweden. We use the same categories as CLIMISH, so it's 1, 2, 3, 4. Also, we didn't want to deviate too much away from the CLIMISH system, because that's used in a lot of regulatory frameworks and you don't want to change all that. But we used slightly different descriptions. And we have an evaluation method of 20 criteria for reliability and 13 criteria for relevance. These criteria are based on other evaluation documents, our practical experience, and results from our ring test. And we also provide extensive guidance in that peer-reviewed document. What's also important to note is that OECD and GLP is not mandatory for reliability. So an example of CRED criteria for reliability, these are the criteria for statistical design and biological response. I'm not going to read them all, but it just shows you what kind of criteria you should think about when evaluating a study on statistical design. So it's about the methods, but also about the results. And this is all taken up in an Excel table that you can use to score the results. And that Excel table is with the supplemental information with the article. So you can just get that on the internet and use it. We first, of course, we designed CRED and the criteria, and then we tested the system with 75 participants from 35 institutions in a lot of different countries, where Australia, regretfully, was not part of it. The participants were, on average, had 7 years of experience in environmental risk assessment, and we looked for these participants mainly during CTEC conferences, gave presentations on what we wanted to do, and asked people to be part of this. They all had to evaluate 2 ecotoxicity studies from a set of 8. In the first round, they evaluated an ecotoxicity study using the Klimisch method, and then in the second round, they used the CRED method. And for 5 out of 8 of these studies, the evaluation results didn't differ if people used CRED or if people used Klimisch, which was good because we want CRED to start to replace Klimisch, but don't want to tell these frameworks that all these previous assessments using Klimisch method, can be put in the garbage bin. No, you want them to be still usable. So for 5 out of 8 studies, there's no difference in the evaluation results. For 3 studies, there was a difference, and I'll show you why for one study specifically. The CRED checklist system with all these criteria shows that the reviewers are more carefully checking all the criteria than they did when doing Klimisch. With Klimisch, it's just like you read the paper and then you have an idea about how reliable it is and you give a score. With CRED, you're forced to go through all these criteria. And then in this study, the problems with solubility limits were detected using CRED, but not with Klimisch. So the study, the people that evaluated the study using the Klimisch method, they all gave the study reliability category 1 or 2. So they said it's reliable. When they used the CRED system, more than half of the participants said the study is not reliable because of problems with the solubility limits. So you can see that the CRED system here, in this case, people did notice the flaw in the study a lot easier. And what you also can see is that, of course, there's still some variability in how people assess the study and the reliability of that study. The distinction between the categories 1 and 3, do we have all information and is it reliable or not reliable? And the category 4, not assignable, is also easier when using CRED. And another example, but not in a graph, is that lack of information on a solvent was detected when people used the CRED system, but not with Klimisch. The key here is that because CRED is transparent and systematic, the points of discussion regarding the reliability of a study are much more easy to identify and report, and then also to discuss. So if there's an issue with a solvent or with a solubility limit, then it's reported. It's not reliable because of issues with the solvent. With Klimisch, it's not reliable. And then you don't know why someone thinks it's not reliable. I also want to show you this tool. This is a web-based tool, the CRED tool, and the evaluation tool for aquatic ecotoxicity. They've got a number of evaluation tools in there. The aquatic ecotoxicity evaluation tool uses the CRED criteria. It's also set up by one of our co-authors. The results of the CRED tool give you a color code. And then here I'd like to ask you, do you think this study is reliable? So it scores all green, except for 50% red on test setup. The study is reliable. The red code here was only for GLP and OECD criteria. So the study was not performed according to GLP. The study was not performed according to an OECD protocol, but that's not a reason to say the study is not reliable. So my message here is that you always need expert judgment. Don't rely on color codes or numerical outputs. Have a good look at what it's telling you. So how are we going to move on now? What we're currently doing is we're trying to develop a CRED assessment course. for different purposes, the Premier project that was already mentioned, but also for CTEC study evaluators, also for authors, the pharmaceutical regulatory framework, and the plant protection products should be available somewhere this year, I hope. And we're doing that together with the UBA and the Dutch Plant Products Authorization Board, the CTGB. And what we're doing is like making different introduction blocks for these different purposes, but then the general presentations on the CRED criteria can be the same regardless of what you're using it for. And then some wrap-up is then again specific for the purpose of the assessment. And we're now working on that middle block of all these different presentations. How has CRED landed in the world? So CRED was designed for aquatic ecotoxicity studies. There's been now also a CRED system for other standard studies for plant protection products and biocides that has been made by the Dutch CTGB. So that's the Pesticide and Biocide Authorization Board, but that has not been published yet. There's also a CRED system for other standard studies for pharmaceuticals made by the German Umweltbundesamt, our colleagues, but that's regretfully also not been published yet, although they are planning to do so. The CRED system is used in a number of regulatory frameworks already. It's used, for instance, very specifically for the derivation of environmental quality standards in the Water Framework Directive in the EU. And it's also the new evaluation method in the new guideline for the pharmaceutical marketing authorization. And there's also been a nano CRED system for nanomaterials published. Work in progress currently at CTEC. There's a group that last week has had a workshop on how to evaluate monitoring studies. And they that second E is not a mistake. They're working on a CRED system. REVM is together with some German colleagues and French colleagues working for EFSA, the European Food and Safety Authority that deals with the plant protection products, to make a CRED-like system to evaluate non-standard studies like toxicity studies for bees, COSM studies, field studies. And within the Premier project, we're currently working on a system to evaluate PNECs or environmental risk limits, because there's lots of PNECs out there that are derived in different ways based on different data that could also use a reliability stamp. The CRED method, it was the criteria for reporting and evaluating. I've been mainly talking about the evaluation part. I want to make a few last remarks also on reporting. So when you have that guidance in that paper on how to evaluate studies, you can also use that guidance on how to set up studies. What is important? What data should I at least report? So we use those same criteria to draft also reporting criteria. And that's important for the students, the PhD students, the master students, postdocs in the audience that are doing their laboratory work. These reporting criteria are divided into 6 areas: the general information needed about a compound, the test setup, the test compound, test organisms, exposure conditions, and the statistical design and your biological response. And as I said before, the advantages of better reporting are numerous. So better reported papers is something that I, as a regulator, would really like to see, because I can then use that information. But also for the authors and the reviewers and the editors, it's very good if scientific papers have all the information already when it's first submitted on the reliability of the paper and of the test performed. So within CTEC, the CRED system has landed into a technical issue paper, and that paper is on the left here. You can find it on the CTEC website with recommended minimum reporting information for environmental toxicity studies. So those are the minimum information that CTEC feels should be in any toxicity study. And I think better reporting is the key for the future, because what we saw at during the CRED ring test is that better reporting does improve the assessment of the reliability, and that recommended minimum reporting information, we feel, is now still a recommendation. And I think at CTAC, with their own journals, that should move to requirements within these journals. Don't publish if this information is not there. And transparency, that's a key point that I've mentioned before. I think transparency is essential. So the publication should be transparent, open science, but also the evaluation of the study, how do we feel we can use this, should be transparent and should contain the information. So why do we think it's reliable or not reliable? So others in other frameworks can use our evaluation or can use also the study, can repeat the study. So that transparency is really essential. Studies should be available and should be evaluated. And that's the end of my talk.